Thank you for the invitation to speak to you today. I get lots of opportunities to give lectures. Many of them are in US universities, some of them are in international fora, but I haven't had the privilege to speak at this institution that I've known about and followed the work of for many decades now, so it's a real pleasure. If nothing else, I think I was selected probably for the variety that you're going to hear from the things you've heard before, and I'm hoping not to disappoint you, because the world that I live in, the world that we practice this type of innovation and company creation, is a rather kind of eclectic corner of the world. And I'm going to hope to kind of expose you to it, maybe infect a few of you, if you're not already doing it, to consider doing it, or if you are interacting with it, to have a little bit of understanding and patience about this breed of people and activity that I'm hoping to describe to you. So with all that as a preamble, let me kind of ask you to just get into the mind frame by thinking about what you would consider in the last, say, 50 years, the most significant innovation in terms of economic value creation and societal value creation. And as you think about it, I've kind of tried to seed the list with a few things that come to my mind. You might have better ones. Certainly the personal computer is probably an important advance that we've made in that time frame. Mobile devices, internet, what it is the internet's allowing us to do in terms of connecting, satellites, the list can go on. In the life sciences, we certainly in the last 30, 40 years have seen the advent of a whole new breed of, or type of therapeutic, biotherapeutics, which now accounts for a substantial portion of new drugs that are being approved. And we've also seen a lot of technologies, many of which are being developed and being approved upon here in the whole measurement world, say the genome sequence being the most kind of, let's say, deserving of attention in terms of what it did as a real feat of humanity. So I may be missing some. I'm not going to ask you to add to the list, although afterwards I'm happy to receive additions. But I would argue to you just for tonight that you think about things a little differently and consider the other innovation that's really come to life in the last 50 years that I would say has a much bigger impact than all of these. And that is the startup venture. Now, what is a startup venture? It is an entity that mere mortals, like you and I, can create with nothing more than an idea and an ocean, and attract capital, resources, be allowed in a legal system to create value from nothing and keep what you create. All of these things are fairly new phenomena. So all of you who follow startups might think this has been going on forever, but I can tell you when I was at MIT in the mid-'80s, there was maybe one company every year of any note that was being created. In hindsight, people describe some of these hotbeds of innovation as creating thousands of companies already for the last few years, but actually this is happening in the last 15, 20 years. And why I'm kind of emphasizing it is because I'm going to end my talk in suggesting to you that we're living in a time when this activity, this kind of innovation, this kind of economic and scientific activity is about to go through a phase change, and that it will no longer be an ad hoc, random, somewhat romantic thing that people with attention deficit and are unemployable do. That's my thesis. And if I'm right, then you no longer have to join competitions and take a class here and there, but you actually should think about a career being a professional entrepreneur, a person who can take nothing and create value out of it in the form of something that when it grows can become the next Amgen and Genentech and Google and Microsoft, etc. For every one of the answers you thought of in the left side, I get to count all of them because they were the product of a startup. And that innovation, I'd say society pays relatively little attention to, more so than the output of that innovation, which is all of the things that we know well. So you can leave now because that's basically what I'm going to talk in more detail about. But give me a chance to tell you a little bit more about this. So my talk is going to basically be in two parts. First, I want to give you a sense of some of the learnings that I've had and some of the things that we've advanced upon in terms of how we think about doing startups in a systematic way and innovation. And then I'm going to use three examples that are related to types of things that go on here at Imperial College in the labs to give you illustrations of companies that came about through this type of process that you might not have heard of yet, and then I'll conclude in trying to bring all this together. Let me just start on this to get you even further in the mood. This is kind of a favorite cartoon of mine, basically describing what it is to start one of these companies. And the caption reads, "This could be the discovery of the century," depending, of course, on how far down it goes. And this is kind of the world that startups occupy. Every idea looks like it's going to cure cancer. It's going to allow mobility, of communication, of information. All these wonderful things, except most of them end up being disappointments. And so it really captures this optimism that often precedes massive disappointment, and yet thousands of companies are being started every month now, as opposed to the few that trickled out. So this is kind of a starting point. Now, to get you further into this, and although those of you who came to hear a healthcare talk, you're going to hear a lot of it, so don't think you're going to hear about only this, but I'm trying to get you into this way of this corner of the world. Another thing is kind of, we all talk about this word entrepreneur. I don't know how many of you stopped and thought about the origin of the word. It's a French word. It comes from the French word for the meaning of which is to undertake. In fact, literally, it means undertaker. If you just translated the word entrepreneur, it's somebody who undertakes. It's quite ironic that it also has the other meaning of undertaker, because quite often you're presiding over what ends up being a disappointment, a failure. But I'm going to come back to this word entrepreneur a little later in the talk. That's why I'm putting this as a starting point, because again, I think there's a lot that people don't appreciate and think about how it is that all these things are happening, and we're learning more and more about them as we have more cases to study. In my case, I did a PhD at MIT in the mid-'80s in the then emerging field at the interface of biology and engineering. And with that, ended up starting a company, quite by chance. And this company ended up being one of the largest instrumentation companies, a company that first launched, for example, time-of-flight mass spectrometry into the commercial markets, which made a big impact on biotechnology, among others. And from there, my career followed a path of a number of other startups. I'm not going to tell you all the details of these. The reason I'm saying it is that I followed the path of most other entrepreneurs. I ended up doing one thing, then that one started taking off. I ended up creating another company, worked with people to start another one. And so far, that's kind of not a very different thing. Now, the 90s was an interesting time because this was all even newer than it is today. And these companies, across many different areas, ended up actually meeting some pretty successful end results. Some went public, some were sold, etc. And I found myself kind of thinking, well, is this really a random walk? Or is there a way we could think about this process? And that's kind of what I want to tell you about today. So Flagship Ventures, which is the firm that this all resulted in, is really an experiment that I'm going to describe to you today in asking the question, can you think systematically about innovation and entrepreneurship instead of doing it kind of, again, when it comes to you or when an opportunity is presented? And I will argue that there may be a way to think about it there, or at least there's some evidence we're gathering to suggest that. So, in a nutshell, just to get kind of the basis for the work I'm going to show you out of the way, Flagship Ventures, on the one hand, is an early-stage venture capital firm, but the part I'm going to describe to you today is this part called Venture Labs, which really is an institutional attempt, that is, with a team, a process, metrics, and learning cycles, to create first-of-their-kind companies around technologies that are developed to solve a problem, that are not developed based on just the advancement of science, but are created to be able to solve a problem that hopefully matters broadly, and to be the first such effort to do so in terms of company creation. And I'm going to tell you a little bit more about that. If nothing else, I think it'll maybe stir some thoughts in how you think about this area. While we haven't done any systematic analysis of what we've done, what you're looking at is simply the names of companies that have come out of this process. There's 27 of them now that have been born in this way by a same group of people working with a broad network of external collaborators. And I'm going to feature 3 of them as we go forward. But if I can just take all this and abstract it into one slide, the things I want to kind of start out with is to make a few observations. Again, if you work in the startup world, this might resonate. If you and I'm happy to kind of discuss how applicable these are based on your experiences. But having kind of spent half my career doing entrepreneurship in a kind of, I'd call, traditional way, and half trying to do it in this more systematic way, there's a few things that I've taken away I want to start the discussion with. One is this strong belief that I've developed, and many others have, that startups and innovations that are basically the founding of a startup are emergent. They are not things that people necessarily design and kind of arrive at in step by step, but they actually emerge. They emerge much closer to, through a Darwinian process, than the stage gate innovation models we think about when we think about innovation. So by emergent, I really mean kind of variation, selection, and iteration. And I'm going to come back to that. Can we think about that more formally, or is that simply things that we observe in nature? And how would it change what we do, actually? The second thing is the slide I referred to earlier, that in this type of approach, you're often working on things, those letters together can't be pronounced easily, but that things that are not yet known not to work. And so the question is, how can you deploy resources and excitement and tenacity knowing that most likely a few years later you may or may not have something to build from it? And what does that do to your decision-making process? How, most people in that kind of uncertainty freeze up. And yet startups have a way to actually move forward despite the fact that there's no objective evidence that this is going to work. Another thing that I've kind of would ask you to consider is this difference between, if you're doing something from scratch for the first time and the world hasn't done this before, how do you know where to get to and how to get there? It's a very kind of difficult decision-making environment. And often people will argue, well, let's go this way, or let's raise some money and go that way. And what I've observed people who do this multiple times end up developing is a sense for a destination. So you've really got to kind of think about these voyages through a series of destinations that you can argue about, should we go to this destination or that destination, and that then becomes the hypothesis towards which you choose your direction. Again, as you sit here, this might seem, but of course, it's just not what people do often in companies, because there's just too much uncertainty, and they say, well, how do I know which destination to pick? And that, again, ends up kind of causing things to be very difficult to make progress in. You've heard, I'm sure, a lot as people talk about startups that people are kind of the most important determinant of failure, and therefore, but deductively, of success. And so how one brings about a group of people that have very different backgrounds, very different cultural adaptations to what it is they were doing before, to effectively create a new culture, and that culture, how it deals with all sorts of pretty profound things like degree of openness, what it is you reward, degree of competitiveness versus collaboration. This is all things that every single startup company ends up recreating. And how to think about that systematically is an interesting area. And finally, kind of this whole notion of survivalism, which accompanies the whole emergent process. You often find yourself unable to win, but you often find ways not to lose. And that then opens up new opportunities. So this is kind of 27 years of doing this in a single slide. And you might say, well, what does that suggest to do differently? And that's what I'm hoping to kind of talk to you about next. So, transitioning to thinking about this in the broader scheme of things. Obviously, startup innovation, which I argued is kind of the big new innovation of our times, obviously I'm biased, is but one of multiple places where innovation happens. And by the way, I won't go into this much today, but in my mind, innovation and invention are two different things. A lot of times people use the word innovation when they really mean invention. My simple definition is that if you could tell me who the customer of that invention is and what value they will derive, then you're talking about an innovation. Most times you can't. You start describing many different customers with many different value propositions, and that basically represents potential, but doesn't represent an innovation. So the act of taking things from either technical possibility or a market need and defining the elements that consist of some technology advance, some form of a product or a service, some value proposition, and some way of protecting, that's what entrepreneurs do in their way. Corporations do it quite a different way. And you see academia on this slide because academia obviously wants to do more and more and more innovation because there's a lot of value attributed to innovation. In invention, you get to license. In innovation, you get to get a part of the value, some of the value that you've enabled society to get. So, and what this slide does, and forgive me for pulling this from different aspects of my life, this is more on the academic side, is just kind of says they couldn't be more different. And that's kind of what I want you to take away from it. And yet, all these different modes of innovation coexist and occasionally have to interact. So a little more specifically, obviously on the corporate side, decision-making, accountability, planning, resource allocation, very, very different world than in a startup where, as I mentioned earlier, you often don't have a culture, let alone a decision-making process. And reward systems are very much built on what value you can create as opposed to a prefixed system. So now, these all have strengths, these all have weaknesses. I'm not here to argue one is better than the other. What I would argue is that the corporate innovation mindset has one huge advantage, and that is over the years, corporations have basically developed ways to make it a bit more predictable. Stage gate processes, metrics for learning, organizational learning, these are all things that I think are huge advantages when you're a Procter & Gamble or a Merck, or I mean, I can pick lots of companies, and I'm not giving the most Airbus, which is not at all available in the startup world. So that kind of mindset has gotten at least some of us, and this is kind of what I'm going to show you next, to start asking the question, could we think about entrepreneurial innovation in an institutional way? Now, I'm going through this quickly because I want to get to some slides on science, so forgive me for just kind of driving through it. Now, I spent some time today here, and I must say I was somewhat ignorant of all that's going on here. And I must say that a bunch of what I heard today are elements of institutionalizing the entrepreneurial innovation process. And I'm very optimistic to see that. There aren't that many places. I teach at MIT, I hang around a lot at Stanford and at different places. And while there are extremes of entrepreneurship you will see and extremes of innovation, there's very little institutionally that does those things simultaneously. And I think we've got a ways to go there. That's an area of keen interest to me. And this is kind of our approach to it. So what we've tried to do is to say, okay, if you're going to have an institution do this, as opposed to an individual project by project activity, then you need to think a lot about process. You need to think about what kind of team can produce this in a repetitive fashion. And I'm going to give you a quick sense of this. Don't expect very much profound, but just at a high level, more to kind of spur thinking on your side than, frankly, this may not be the only way of doing it, but here's an experiment anyway. And the key is that we've been doing it this way for the last 14 years, which means we've iterated quite a bit how we do what we do. And 26, it says on this slide, the 27th company came out of this process about a month ago. And in aggregate, these companies that were born in this way within Flagship's Venture Labs unit have attracted well over $1 billion of capital, have created about 300 patents at their founding. These were the patents that came out of this innovation process. And a number of them have introduced products or have products in the clinic that I think can be measured and tracked. So what is it that I'm trying to talk about here is I think that to think about this in a systematic way, you need to think of it on the one hand as innovation and on the other hand as entrepreneurship simultaneously. And the process on innovation, as I mentioned, has many different varieties, but you need to be kind of on the extreme of both. There's lots of approaches that are being taken that are various variations on this theme, and I'd say kind of pushing to the outer extreme is the challenge that we've and others are trying to begin to take on. So how do we go about doing this? A couple of more slides of this type, and then let me give you instances. So one is, is that we don't one thing you have to resist to do is to take your first good idea and run out and raise money, because God forbid you actually get money, you now have to actually do that for the next few years of your life. And long before any of us have a chance to think about, well, is that the best thing, that version of what we could have done? Do we have any evidence that actually that can create the kind of value we're purporting? You just rush and create the company. Certainly, again, where I come from, where there's a very sophisticated, multi-hundred financing firm ecosystem, it's become a bit of a game of having an idea and some support, and then off you go to the races. We think differently in that we think a good way to start exploring a new area, and I must say I'm describing this in areas where there are no companies yet, so kind of an emerging scientific area. The microbiome, for example, several years ago, was such an area. Cancer metabolism would have been such an area a few years ago. It still is in some ways. New forms of therapeutics, new types of approaches to perturb biological systems. These are areas where you say, well, what do I work on? And if you don't have a good answer, the first thing we would suggest one does is really to come up with a range of hypothetical ventures that you could actually define, extrapolate from things that you can cobble together, and describe initially just on paper. And until you do that, I would argue that any one idea you work on is going to be a local optimum, and later you'll find out that there might have been something more interesting, but now you're heavily down the path of developing that one thing. I'm not telling you anything profound, but taken together, I can tell you that it ends up being quite a different approach to doing this. Second is that if you believe, as we do, that these things are going to emerge, you want to get a lot of engagement from folks, and we have our own networks, you have networks here, to help in the selection function, right? So you can be managing variation. So the hypothesis generation is think of it in a Darwinian way as variation. The selection function isn't just about you sitting there as an investor or you sitting there as an expert in an area. Selection gets huge benefit from a crowd. And on that basis, I will tell you kind of one deep insight, I'm only half joking here, that I would propose you think about, because you're living in a place where this will make sense, I think. And that is, I would argue that experientially, the second most abundant resource, renewable, free resource on the planet, the first one you would say is solar energy, abundant, free, renewable. I'd say the second is people's propensity to tell you how stupid your idea is, if I can be blunt. It's renewable, it's abundant, it's free. It is. And in a highly educated environment, it's an extreme sport. I mean, people will be very glad to tell you that's just kind of what we all do in academic environments and companies that are dominant in their space. Well, I would just invite you as potentially an entrepreneur who's thinking about this to use that resource. Because if you go to somebody and ask them for a good idea, my experience is they will ask you to pay for it. Why would they give you their good idea for free? If you go to them with your idea that may not be fully baked yet and start engaging them in a dialogue that might help shape the idea, what you're going to find is a significant amount of crowd-sourced input that at the end of the day, as long as you're humble enough to act on their input and technically experienced enough that you know what to vary to overcome people's objections, what will emerge, I can tell you this is kind of what we do for a living, what will emerge is rather unusual, not a linear extrapolation from what preceded it, and could be the basis for an interesting venture. Now, at the end of that, all you've got is something I would argue you should prototype. Now, I'm not here to tell you for the first time what a prototype is, but I would argue, ironically, that other than everything else we ever make just about today in products, people don't generally prototype companies. Right? Nobody really stops and says, you know what, I'm going to go down this path and then I'm going to shut it down voluntarily. And the reason for that is that if you're an entrepreneur, shutting down a company is unemployment. It's a kind of a counter kind of instinctual thing to do. I'd like to believe, and many of us who've done this many times, serial entrepreneurs learn how not to get stuck working on the wrong problem, but a lot of resources wasted because these things are not prototypes. So I would say think of a company before you launch a new company, and think of it as a proto company. Something that wants to be a company, but that you're trying to front load tough, killer questions. Questions that actually could liberate you from working on the wrong problem. Now, you might say, well, how do you know? And how do you know you didn't miss one? All I can tell you is it's sure better than not doing it at all. And I would argue that very little of this actually happens. It happens by default because companies tend not to raise money and they will have become a prototype in hindsight. By the way, a lot of those prototypes might have been good ones. That's not a good way to do prototyping. So the second phase of what we tend to do is kind of go through this prototyping process of a few, and only then will we actually launch a new company. And you could imagine then that these themes that are on this slide, which I will not spend a lot of time on, of rapid prototyping, customer immersion, these are things that become now the elements of the culture I would argue it takes to think systematically about disruptive innovations and company creation. You need to have an ecosystem that is rich with many different points of view from other entrepreneurs, points of view from academia, points of view from institutional research, and points of view from large companies. Again, I would test, you should test my hypothesis, take an idea that you haven't heard the equivalent of, expose it to someone you know in a large company, and see if they give you a useful feedback on it that can give you the next suggested step without any consulting agreement, without anything. Now, you might be afraid of IP, but there you have to kind of develop a way to describe the idea in a way that isn't the basis for IP, because at the end of the day, if your patent is on an idea, we can have a separate discussion. It's not going to be a very valid patent anyway. You're going to have to do more than just have an idea for a patent. So this is kind of the approach. We've applied this approach across a whole lot, I don't expect you to read this, of different scientific areas, totally different types of output companies. And all I can tell you, at least, is this many years of doing it later, it does appear that some of this lends itself to learning cycles and to making it systematic. Which brings me to this point. This is kind of the portfolio of companies that we've developed over the years in this way. And what I thought I would do in the next bit, which will be quite a departure from this because it'll be more about hopefully new technologies and new innovations you will hear about, is to focus on three of these. Moderna, which has developed what I would argue is a completely new way of introducing therapeutics to patients. The second is Pronutria, which is really operating at the interface between nutrition and health, but more importantly, molecular nutrition and health. And then the third, which is, maybe all of these are close to what people are doing here, but I know for one, because I met with Professor Nicholson earlier today, certainly the microbiome area where we've been active for the last 6, 7 years, and most recently have worked on a particular company that came after many, many proto companies we had developed. So let me do these in 3 little vignettes, and then I'll wrap up, see if you have any questions. So one of the explorations we took on in 2009, 2010, is to ask whether there could be completely different modalities of drugs that we have not as a industry explored before. You might say, why would you care about that? Well, if you've worked in this industry for many years, as some of my colleagues and I have, you'll realize that small molecule drugs and proteins occupy 99% of what goes on, vaccines a little bit, and gene therapy kind of made an attempt to be relevant. People are still working a little bit on it, hasn't really had critical mass. Antisense siRNA, another attempt to do things with RNA, getting there slowly. And we recognize that any of these types of fundamentally new approaches end up being quite interesting if you come up with it, because you can come up with many, many drugs out of a whole class. And so we've been interested in that. We've looked many different ways. We've worked, for example, many years ago on one of the earliest forms of antibody mimics. So antibody being drug category, we worked on what are called adnectins. These were fibronectin domains that mimic what antibodies do. So there's a lot of reason to care about this. It's kind of, take my word for it if you're not in this area. Pharma companies might be looking for the next drug, but they're equally looking for a whole new category of drugs because they know how to make many drugs out of that. So that was the kind of motivation. And we looked at lots of different approaches to lots of different molecules. And we kind of became interested in this question, which is, why is it that mRNA is not the basis for a drug today? Right? So those of you who work in molecular biology and pharmaceutical, it's interesting. We have drugs out of proteins. People have tried drugs based on DNA, and people have done vaccines that are commercial with DNA. But mRNA, kind of as a category, isn't available to basically perturb human biology. And the reason for that, those of you who work in any aspect of the immune system know, is that if you introduce mRNA into a mammalian cell, you will get a massive innate immune response. All hell will break loose. You will get interferon. You'll have protein synthesis being shut down. There's a huge innate response to mRNA because basically cells see this as a viral infection. Now, as we looked at, okay, well, how can you overcome that? With all the chemistry that was being done to modify RNA for antisense, some of the work had spilled over to actually find other modifications that might attenuate the immune response of RNA in general. And in particular, some of the work suggested there could be some modifications that could have less of an innate immune response to mRNA. None of it was getting rid of it completely, and it was being used when we saw it to use Yamanaka factors, these factors that take skin cells and convert them into stem cells, to deliver them through mRNA by these modifications. What interested us, though, isn't that at all. That was a technology kind of bit, but really was, what would it take, and could we actually prototype and de-risk the notion that if some modifications did some of this attenuation, could we find others that could completely eliminate the innate immune response to mRNA? Now, I'm putting a lot of work into kind of a few sentences, so forgive me for that. And on that basis, we actually started doing a systematic analoging of the kind of canonical bases that would make RNA to see if there are variants that actually have a much stronger effect. And about a year of this experimental work later, we found some that indeed did. And that formed the basis of what I'll describe to you next. And again, I don't know if any of you have heard of Moderna. Probably not. It's a little company in Cambridge, Massachusetts. But in a short time frame, since 2011, when our experimentation fully started in a company setting, let me tell you what we've been doing. So the goal here is to make modified mRNA that is the drug. It has a cap, it has a tail that attenuates translation, it attenuates the half-life of these things. But the goal ultimately is to make a molecule that you could stick into a patient and have a patient make their own drug, make their own protein. That protein could be factor VIII, that protein could be insulin, that protein could be, ideally. So all of what we think about through recombinant DNA technology, which is a way to get E. coli or a yeast or hybridoma or CHO cells to make a protein, every cell in your body knows how to make a protein. If only you could get to tell it what protein you wanted to make, transiently, because otherwise you would be doing gene therapy where the viral incorporation of the DNA and its permanence could cause safety issues, as it has over many years, and people are slowly beginning to address. So what is the game plan then? The game plan would be to make messenger RNA into a therapeutic, administer it, and then, and I'll say this, in this case, we knew that if we could do this, you could not only make proteins that are secreted, but also proteins that are intracellular, because the mRNA gets into cells, a subset of it does, and if the protein doesn't have a secretion signal, it'll stay in the cell. So it's kind of an interesting thought. You probably are sitting here saying, no way will it ever work. And you may be still right. We tried it in mice. We made these things synthetically. By the way, it takes a week to make an mRNA suitable to inject into an animal. We order the DNA. We do a little bit of the engineering. We've learned how to do that much better nowadays. And we formulate it in saline. We formulate it in some liposome. We may formulate it in something more sophisticated, depending on if we want to do intramuscular or IV. And we tried it. So this is kind of very little bits I'll show you. This was mRNA for EPO injected into intramuscularly, I think, in mice, although I should just double check. Yeah, it's intramuscularly, biweekly injection. 8th dose out, you're making EPO in the blue compared to a vehicle. If you had an innate immune response, that would shut down pretty rapidly. Within a day, you wouldn't make any protein. And we've done lots of that. I can tell you there's lots of mRNA we've made, modified and not modified, that just completely gets shut down. The second question was, can you see dose effects? So can you actually put less in and more in and see somewhat of a difference? In this case, we're not just measuring the amount of the EPO made, but we're also measuring the effect of it. So this is a hematocrit count. And so we saw some encouraging data. This is about a couple years old. Then the question is, so lots of diseases have been cured in mice. We all know that. What would happen if we actually went to larger animals? So within about a year of starting, we tested these in non-human primates. And what we found is if you adjust for the volume of blood, we basically get the same amount of EPO made in each of these species. And the dotted line here is now showing the serum levels in human. This is just EPO. So you might say, yeah, but you got lucky with EPO. I'll spoil the fun and tell you we've now made about 300 different mRNAs for 300 different proteins that are either in clinical trials or on the market. There are some exceptions to things that need to work in very precise levels. But other than that, if you're trying to have gain of function and introduce a new amount of a protein, that we have not seen a limitation in our ability to do that, at least in animals. This opens up some interesting new possibilities. One of our collaborators and founding academic, a scientist, published very recently, Ken Chin, who's now at the Karolinska, used to be at Mass General Hospital, published this work. And I don't have the time to go through in a lot of detail, but let me just tell you what the experiment is. This is VEGF mRNA injected once at the site of an infarct in a mouse. The result is a dramatic survival increase. You basically, in this case, are getting blood vessel formation almost as early as we can measure it. And if you kind of look at the data, this has been published in Nature Biotech. I invite you to look at it. This actually, so on the top right side is the imaging of the vehicle treated versus the VEGF treated. It's a single injection, and so now it's actually been reproduced in pigs as well. There's a lot of questions this raises. What do you go after? Do you do rare genetic disorders where you're lacking something that you want to make more of? Do you go after cancer where there's interesting new targets? You're not going to hear any more about it because I'm limiting it to a little vignette, but I'm happy to talk to you afterwards. This kind of technology and the experiment, I don't know if one could have gotten grant funding to do all this in this time frame, only because the scientific underpinnings simply weren't there when we started. We didn't know if it was going to work, except for about $1 million we could do the experiments needed to find out if it was going to work, or at least work enough to warrant what has now become significantly additional funding. So let me kind of tell you roughly what has resulted. What has resulted is that this company that was conceived in 2010, was in a lab in the middle of 11. In 2012, after 18 months of actually filing some intellectual property and beginning to do some work, we kind of announced the company's existence. In 2013, we did one important large partnership with AstraZeneca. AstraZeneca took on the rights to up to 40 drug molecules from this platform over the next 5 years in cardiometabolic disease and in part in oncology. Then another company in the rare disease area, based on data we had generated, did another partnership. Combined, these two partnerships provided the company with about $500 million of capital, none of which is going to develop these drugs, all of which is developing the platform and other drugs. And obviously money is not the only measure of early success, but if you don't have the capital, you're not going to build this platform. And we're off now. The company's about 150 people, all scientists, and we've built a pretty interesting a team to go after applications of this. So it's an example, but I'm tying it back to the way it came about, because this is not the result of an invention or a long-term research program. This kind of is an emergent kind of outcome of asking some hypothetical questions. I want to give you a couple of other examples. I'll do them much more briefly because we're running out of time. Let me tell you a little bit about another company you probably won't know anything about, called Pronutria. Pronutria is trying to create another new class of drugs, if I can offend you with that notion, because I know that people don't create new classes of drugs every day, but we're trying little experiments to do it. We really believe that there's something to be said about innovating in the drug class as opposed to within a class. In this case, the idea was the following. We took, for reasons that I won't give you the history of, we took the human diet and basically disintegrated it into a proteome library of the proteins we could identify in as many foods as we could get our hands on. We did this through LC-MS/MS, we did this through informatic genome sequencing-based inference, and we created a protein database and a peptide database well in excess of 1 billion constituents. Every one of these, we know which food it comes from. So you might say, so what, right? One, it's quite remarkable that there's well over 1 billion proteins our bodies interact with at any given time through our diet. But that diversity has never really been looked at as a source of pharmacology, right? So we've looked at natural products, but these are natural products that we kind of co-evolve with because we eat them. And just like the next example I'll give you, which is the microbiome, these are things we've co-evolved with and vice versa. And so the question we wanted to ask is it possible that there are just 1 or 2 more examples of a category that we suggest we call these pharmacol nutrients? Why is that? Well, those, again, you may be experts in this area, so sorry for the introductory slide here, but there are 2 really good examples of things from the natural diet. One is omega-3 fatty acids, that is the basis of Lovaza, a $1 billion drug. The other is niacin, which is the basis of a $500 million drug, Niaspan. These were both food-derived nutrients that had pharmacological effect, or at least people did clinical trials to demonstrate them at the right correct dose, and had an approval as a reimbursed drug. And our notion was, is it possible that there are such agents in the diet, in the human diet, that are proteins instead of fatty acids or vitamins? And that's kind of what we've been going after. So let me tell you kind of where we are. What we're trying to do is piggyback off of decades of amino acid pharmacology that's been done through mixtures of amino acids. People have shown very potent effects on anabolic muscle growth. People have shown effects on metabolism. People have shown effects on cancer, certain dependencies of amino acids. Certainly inborn errors of metabolism, many of them are amino acid dependent. PKU is a good example. There are many others. And all of that work doesn't really feed into anything. You can adjust your diet, but what that work allowed us to do is to actually go into this database and say, well, are there specific proteins that have profiles that might actually have stimulating effects on specific pathways? And that's work that we've undertaken. The interesting parenthetical thing I'll mention to you is that because these agents are starting out from food, they are in a different category of compound than a new chemical entity. These are things that you can demonstrate through safety studies that they could fall under the category of things that are generally recognized as safe. Grass, because they are being derived from food. This is not the first time your body is seeing these. Effectively, it's a drug delivery vehicle for amino acids. So it's a prodrug, if you think about it that way. And so that means that we have the ability to test them in humans as a potential supplement or a medical food long before we have to decide what formulation and what dose we can develop them as drugs. And so we have actually begun to do this, doing the right safety studies and then testing them in humans after doing animal work. There's a whole lot of candidates that we have. The most advanced are in muscle. And in particular, and again, in the interest of time, I'm going to skip this, so sorry for flying through this. In muscle, there's a lot of work, published work done, connecting leucine-rich and in general, branched-chain amino acid-rich amino acid content to activation in the mTOR pathway. That in turn, we have reproduced preliminarily already in human data with proteins, not amino acid mixtures, and that's a very different delivery form. This slide kind of summarizes all this, and let me just direct your attention to a few things. One, on the top left, we have many, many, many different proteins from many different sources that we could choose from that have an interesting composition, first of all. So in the drug discovery space, as you know, the more shots on goal you have, the more you might miss with a few, but you still have a candidate. Here, there's no shortage of candidates that fit these profiles. In other words, amino acid composition profile. Second, we're interested in things that will digest quickly. Whey protein, which is one of the 5 waste-derived proteins that are used for muscle health and, for that matter, nutrition applications, digests, half-life, if I'm not mistaken, is on the order of 20 minutes. We have proteins that will fully become amino acids if you ingest them within half a minute to a minute. So digestibility is another parameter you can choose for. Thermostability is a parameter you could choose for. These are proteins you'd rather just kind of keep in room temperature or elevated temperatures and have them not kind of auto-degrade. That is an important factor. And then finally, again, things you might be, I'm inviting you to think along with me. This is not, we're not selling you any of these because there's none of these on the market. But the thing to think about is how to think about this a bit out of the box. If you want to take enough protein to meaningfully affect muscle health, especially in an elderly population, the amino acid data suggests that you'd need to take 4 bottles of Ensure a couple of times a day to get enough protein to do this. The solubility of the protein that goes into Ensure then limits how much volume you need to digest, or dictates. Well, in our case, we actually have some proteins. The most advanced programs is PN107 that has solubility of 400 grams per liter, right? As opposed to 10 grams per liter, which is kind of for a typical protein that's formulated. So these kind of open up new spaces where by no means are we out of this process. We're working in muscle. We're also beginning to work in metabolic diseases. More to come in the future. But again, this is kind of an emergence of an exploration, another exploration at the interface of nutrient health and new modalities. Let me kind of just quickly wrap up. Probably there's people in the room who care a lot about the microbiome. This is the approach we've taken to the microbiome. I think you all know that there's a lot of scientific publications. In fact, it's been on The Economist and many other popular press, so that's usually a sign that it's almost too late. But anyway, it's certainly an emerging area. More important to us than the publications is really that people are beginning to find correlation and already speculation of potential causation, although I think we have to be very careful about the complexity of the underlying biology before we can make those statements, to many different diseases and the state of the microbiome. Now, I think that's well known. The more interesting thing for us, let me just quickly jump ahead, is the following observations that our kind of research and our exploration produced. One, kind of this notion that our traditional intuition that these things work in ecosystems and they work in symbiosis is borne out when people do additive experiments and they bring elements together and construct microbiomes as opposed to just observe them by sequencing. There's data that we've developed and others have developed. Second, that we had the hypothesis that in order to perturb a microbiome, one approach might be to create a minimal set of organisms that can actually cause a shift. And I think those of you probably know that the most advanced microbiome-related work that happens in medicine today is fecal transplants. And this kind of came out in the public press only 2-3 years ago. We had been talking to people doing fecal transplants for about 6 years. And what's interesting is when you talk to all these people all around the world, their protocols couldn't be more different. And they all kind of work. And that usually suggests, and at least suggested to us, that that is a much more overspecified system. In other words, you don't need all of those organisms. You probably need a smaller subset. So we set out to find a set of organisms that could recapitulate what a fecal transplant does, except do so in a form we could make into a drug, orally consume, and in restorative, particularly in C. diff. I will spare you the details. Not much is known about this company, by the way. They haven't really said what they do very much, so I'm probably telling you more here than they have. But I'll just very quickly at a high level. There's a computational component to this. There's a combinatorial construction component to this. We try hundreds and hundreds, if not thousands, of combinations of organisms that are selected for their metabolic profile, not their species representation. And we've basically gone down this path to identify a few testable combinations. And again, I apologize, I'm rushing through this. For those of you who care about the details, I'm happy to engage afterwards. But the most important thing I wanted to say is we've ended up going from that starting point of the informatics all the way to several combinations and attempted to show, at least in mouse models, what people have seen with fecal transplants in mouse models. And we've seen the same type of efficacy effects. Probably more relevant, and so this is, I think that's what this is kind of showing. Probably more relevant, though, is what what I don't have slides to show you, which is that we've also then gone to humans. And we've announced earlier this year, and we've done now more studies, we've been involved in testing an oral capsule with a subset of the organisms that is in the human microbiome that we have selected for what we believe to be the effect that you're getting through a fecal transplant. We've treated 27 patients. In one of the 27, we had almost an instantaneous recurrent. They should not have been in the trial. In the other 26, we've seen the same effect that successful fecal transplants show, which is that the recurrence is eliminated over the time frames that people do these studies. These were done at Mayo, at MGH, at Brown, and on the basis of the human data, we're actually now moving towards doing a phase 2/3 type trial, which is being designed to test whether we can actually develop a product based on this approach. Now, this approach for C. diff, we've seen anecdotal data, also has an effect on other pathological organisms that are in patients that we've been treating. So it's having a pretty substantial effect on the microbiome as opposed to a specific C. diff effect. And a lot of research is now going into figuring out whether we can do, kind of address other types of diseases in this approach. We care about this because C. diff, recurring C. diff is a fairly large problem. In the States, certainly, you've got a significant market. In the US alone, you've got about 50,000 relapses and significant mortality. And that's why the FDA, which has expressed concern about this fecal transplant procedure, has not completely discontinued it because it's very effective. And so from this base, we are looking forward to also applying this methodology of creating what we call these ecobiotics, these ecosystem-level combination of organisms in metabolic diseases, and a lot of work going on here and elsewhere in that arena as well. And of course, I'll kind of end this little bit and then give you some conclusory statements. What you can do as a therapeutic, just like in the case of Pronutria, you could also think about it as GRAS agents. So kind of, other than the fact that the word probiotic is used for an ineffective intervention usually, I think that in the future, such things will also be used in the supplement world and in the medical food world to actually have health effects. So there are other development paths to be followed. So let me just wrap up. I know you've kind of suffered through two very different presentations, but let me try to kind of bring it together and give you a couple of parting thoughts. These three examples, and I can give you other ones, are only demonstrations of an innovation process that is exactly what I described earlier. These did not start with a single technology starting point. They emerged. And as we think ahead, and particularly for actually Imperial College, which I learned has a very active entrepreneurship innovation ecosystem, I actually wouldn't mind if the administration also challenges themselves along these lines. And let me just tell you what I mean. I started my talk in saying that I think all this is going to lead, and everything that's going on around the world in this space is going to lead to entrepreneurship becoming a profession. And what I mean by that, and I'm going to spare you all these words, is this. There are other professions that have existed in some of our lifetimes. None of us are 100 years old, but if we went back 100 years, management as a profession didn't exist. Architecture, you can go back and trace its beginnings. Medicine, I'm sure people here are quite knowledgeable about how medicine started. But before medicine became a profession and a discipline, a lot of things were done in ways that were representative of where it came from, which people attributed great astonishment and magical powers to, which over time now has been susceptible to some body of knowledge, some of which you can acquire through learning by doing, some you can learn through actual exposure to content. And I would argue, and you might reject it now, and let's come back and talk 5, 10, 20 years from now, see where we are. But I would say that business schools today, which are absolutely created to teach management and are absolutely doing a great job introducing entrepreneurship and innovation, we can go a long way in thinking about technological entrepreneurship as a discipline in its own right. Some of things are in common, a lot is not. How you do marketing or financial aspects or intellectual property or culture or all of that is very, very different when you start with nothing, I would argue. I may be wrong. And the result I would just challenge all of us to think about is, can we eliminate some of the failures that are not due to the fact that the particular technology didn't work out, but are due to the fact that this is being done by a bunch of people like me who started out as economic hacks. Basically, it's like software hacking. We basically can go raise money, start a company. I did 27 years ago. And a whole lot of it fails. A lot of money is thrown out the door. I think as a society, maybe I'm wrong, but I think as a society, we're at a critical mass of experience that this transition will happen. And why this matters to an institution like here and regions and countries is that if I'm right, the thing to emulate is not Silicon Valley or London or Cambridge, Massachusetts. The thing to emulate is the teaching hospital of the future, the equivalent of that in this area. Is how is it that we will prepare students with technology backgrounds in this particular arena to spend a career of entrepreneuring as opposed to what I did, which is have to wing it at some great cost to society. And on that thought, my last slide, I promise, is this, which is I would ask you to come back to the word we used and just say, as a favor to me, sometime in your life, use this word, see how it feels. We all view this as entrepreneurship. Now, in the English language, when the word ends in ship, it means the state of being something. Leadership, craftsmanship, sportsmanship, all these ships, right? It means the state of being something. Now, I'm an engineer. I don't do engineership. And why is that? Because engineering is the ending ing, is what it is when you're actually doing something. You're not being something. And as a society, we view entrepreneurship as a state. It's if you're an immigrant and if you are kind of impatient and if you're risk-seeking, then you might be a good entrepreneur. All these books are written about it. I must say, having lived my life with it, and I'm surrounded by hundreds of them within our portfolio now as elsewhere, I think that's just because of where it came from. It was kind of nebulous because it happened in an emergent way. But I would argue that what we need to help people figure out how to do is entrepreneuring, not entrepreneurship. And some people are going to be great at it, like great brain surgeons. I don't think medical schools produce great brain surgeons. They produce brain surgeons. Some of them are great. And we can decide what makes them great, dexterity, attention, all that stuff is how good you are, not whether you can do it. And I think that the challenge in thinking about entrepreneuring as a discipline is really, and I obviously use this slide at MIT because I'm trying really hard to convince them at a bastion of what they think they're at the top of the world in entrepreneurship, as the Stanford, to say actually that's got a short half-life and it's going to be taken over by a whole new discipline doesn't make me popular, but that's why I use slides like this. I would also urge you to think about entrepreneuring 10, 20 years from now as an object that people kind of come and be trained and give experience to, and then they do for the rest of their life. So on that note, thanks for your attention. I went a little over, but thank you very much.